Yes, so today I'm going to chat a little bit about the work that I do, which is very wildlife focused. And I was going to start with something that we all know, and that's that we all here understand the concept of One Health. And I think with the fact that 70% of infectious diseases are zoonotic, it really sort of underscores the importance of maintaining wildlife health to protect human health. And we've found a number of instances where this has become apparent, and I think especially for the general public, following the COVID-19 pandemic, this became very apparent for the general public as well. We've recently had a Marburg outbreak in Ethiopia, and at the moment there's a Nipah virus outbreak that's causing havoc at Asian airports. So the thing that we really need to think about is that we need to be able to rapidly detect and respond to wildlife morbidity and mortality events, and that's critical to reducing threats to both wildlife and human populations. And the key for this really is surveillance. So for us to be prepared for future pandemics, we kind of need to have these implemented long-term wildlife surveillance programs, and particularly focusing just on wildlife health. For me, I'm really interested in how parasites affect host behavior, and that this can happen in different ways. So for example, the zoonotic parasite Toxoplasma gondii, it infects rodents, and then the reproductive home is the cat's gut. And so when the rodent gets infected, they change their behavior and they become less afraid of cats, and they are less afraid of cat urine, and so then the cats eat them and the whole cycle continues. So that's interesting about how parasites can affect host behavior. And then the other thing I'm interested in is sickness behavior. So for me, as soon as I get the flu, I just want to crawl up in bed with hot chocolate and Netflix. And you can imagine that wildlife feel the same way, but they don't have those kind of options. Something else that I'm interested in is that when we think about wildlife disease, we usually think about one parasite like rabies, or we think about West Nile virus, but the majority of wildlife have more than one concurrent infection. So they have a number of different co-infections, and these could be having disparate effects on their behavior. So most of the studies that we've done before are these single host, single parasite investigations. So the other thing that I'm interested in is that we have a number of wildlife species, and we know this from being here in Tucson and even Phoenix, but we have a number of species that are well adapted to urban environments. And so I've put the two examples here of bobcats and coyotes. Bobcats are the natural reservoirs for the tick-borne pathogen Cytosine felis, and that causes bobcat fever and is fatal to cats. And this was a recent study out of Canada, where they were looking at coyote scats in cities being an increase of zoonotic disease in North America, and this was particularly for the Echinococcus multilocularis parasite. And they were showing that coyote scats being in the cities was increasing human cases. We also have the issue of things like rabies, bobcats and rabies, and those interactions with people. So having these wildlife in cities does really increase our chances of zoonotic disease transmission. And then one that we're currently working on with Pima County is looking at rodents and hantavirus. So in November of 2024, there was the first locally acquired case in Tucson in over 30 years. And then, of course, there's birds and HPAI and West Nile virus. And so we have things in our gardens, like we have bird baths and bird feeders, and these are all areas where, well, bats and birds will congregate. And so they're nice areas where we can have disease transmission happening. And then you might have neighbors with little ponds or tires lying around that collect water. And so we create these lovely mosquito breeding grounds. And I always like putting this one in because this is an example of how animals can change their behavior based on what we have. And this was a coyote that was on the tarmac in the Portland airport and then just jumped on one of the trains and sat there all happy like a little dog. And people didn't know what to do, but it was completely comfortable around humans. So the more they lose their fear of us, the chances of transmission of zoonotic diseases really does increase. So one of the things with wildlife surveillance is that it's challenging. It's usually very costly. It often involves captures for sample collection. A number of these samples need to be stored in a specific way for them to be viable for the tests that we want to conduct. And doing long-term monitoring, which is like essential for determining trends, requires a big investment in equipment and personnel and data collection. And a lot of the work that we end up doing is very reactive. So we find something going on, we notice all the dead animals, and then we go and start our surveillance program. but it would be really nice if we could do this in a proactive way. And so one of the things I'm interested in is feces. No, that sounds ridiculous. My kids think it's hilarious. But anyway, we look at feces and we're wondering, could microbiomes be the answer? So there's a couple of recent papers that have suggested that it might be a useful tool for monitoring for conservation benefit. And if we think about microbiomes, we know that that's a collection of bacteria, fungi, viruses that are found in and around on the body. And for what we know about this is mostly the work has been done in human systems. As you were saying, we're very human-centric. And we have a lot of animal models, like we've got zebrafish, and we have mouse models that will often be used to test different theories and experiments. There's a lot of work that's done on the gut-brain axis, which for me is really interesting, again, because if your microbiome is different and it affects your behavior, if we have the same thing in wildlife, we could have indications of changes of behavior based on what the microbial signatures look like. So, my thing is, can't we use fecal samples as a non-invasive way of monitoring wildlife health? Because from feces, you just get so many things. You can get microbiomes, you can get the viromes, fungi, you can get parasite information. You can also look at stress levels, and you can determine diet. So those are kind of important, especially if you wanted to look at how diet affects health and how that affects microbiome. If you've got a rural individual versus an urban individual, all of these things can play a role. So some of the work that we're doing for wildlife surveillance in my lab at the moment is we're looking at monitoring avian species for West Nile virus surveillance. We've been working on modeling the mosquito distributions, and now I have a grad student who had a poster up earlier, we're trapping the birds and we're collecting blood samples and swabs, and we're trying to look at how the virus changes over time within the bird species and how that matches with what we're seeing in the insect pools. So we're doing this across Tucson for at least the whole of this year to get all the different seasons, but we're also collecting swabs because we thought we could try and look at microbiomes and see if there's any microbial signatures that match with the competency levels that we're finding in the birds. This is some work that a master's student of mine did. So we got gorilla samples from one of our co-PIs in Uganda. And so mountain gorillas, gorilla barenge, there's only about just over 1,000 individuals remaining. Their population is under a lot of pressure from habitat loss and disease, and so we looked at microbiome and parasite communities from 4 different groups. So this was in the Bwindi impenetrable forest. So you can see there's an orange group there, that's the Kulingo group, and then the blue area at the top was Bahoma, and that's 3 different groups that we were able to get samples from. So these were all groups which were habituated to humans, so they were ones that people went with on tourist things, so we just had a research to go with and collect samples. And we were able to get individual ID from the ones in the blue area, but not from the ones in the orange area. So what we were finding is that some of these gorilla groups would stay in the forest during the time that we were collecting this, and others were venturing out into the local communities and raiding banana trees and things like that, but sharing water sources with the local communities. So we decided that we were going to have a look at, was there a difference between the microbiomes and parasite communities of the gorillas that were in the high proximity areas and the low proximity areas. So you can see I've got Bahoma 1 was a high proximity area, and Kulingo was another high human proximity group. And so we actually did find quite a lot of differences. So the 2 groups on the top there, the red and the green, or the pink and the green, those were the Bahoma groups that stayed within the forest, and then the orange and the blue were the 2 groups that were going outside of the forest into the local community areas. So we could see the separation based on whether or not they were accessing human areas in their microbiomes. And another thing that's happening that's been reported by gorilla doctors within the Uganda system is that they're having this chronic wasting syndrome. It is not chronic wasting disease, they're calling it chronic wasting syndrome, and they think it's related to an infection with a strongylid parasite. And so what we wanted to see was, what does the parasite communities look like, and do they differ in the groups that go into the adjacent communities and the ones that don't? So we found that the ones that were going into the adjacent communities had a much higher parasite diversity, and this was using 18S data, compared to the ones that were not going in and was remaining in the forests. And you can see that the separation when we're looking at the sort of community composition was not as distinct, but you definitely do have that clustering on the left-hand side of the right-hand side graph, where you can see that the groups that were going out of the communities did cluster together. So we also had a look at what the parasites were different if we just split the parasites up and said, were you in the high area or were you not? And we had a whole lot of these Rhabditidia groups for the order, where the groups that were going out of the forest had a much higher number of that than the others. And so that group is the one that contains that stronger lid that is shown with chronic wasting syndrome. So it's an interesting thing. It's not definitive, but it's trending in that direction. Other things that we look at are, we've had a study where we looked at mange and coyotes, and we thought maybe we'll be able to find some sort of bacterial signature linked to mange. And actually we were able to. You can see that there's a whole host of bacteria that were enriched when the species had mange, when the coyotes had mange, and a couple that were depleted. So some good ones were possibly not there, and some ones that probably not as good were there. And the caveat was that we only had a few individuals who actually had mange, so this is just like preliminary data. We're hoping to get more samples to be able to look at this in more detail. We've also done some work on viruses with bobcats. So these are not non-invasively collected samples, but we wanted to look at viruses. So we took swabs with the blue dots are the swabs and the red dots are from the scats. And what you can see here is that some of the viruses are found both in the swabs and the scats, and others are either in the swabs or in the scats. So that's an interesting thing to think of, that we might not be picking up certain things. Okay, it just jumped up. So this Felis puma virus here is one that has been found in domestic cats and pumas before, and then with our project was the first time that it's been found in bobcats. So that's showing a definite transmission between pumas and cats. And this is from the Bobcats in Tucson project. And it's just going by itself now. I wanted to highlight that we're not the only lab that's looking at this. So this is some of the work of the people in the room, Crystal Hepp and Faith Walker. So they've looked at whether they can use bat feces as a means of monitoring rabies, and they've been pretty successful with this. And then there's another instance where it's a very recent paper looking at whether we can look at fecal volatile organic compounds as profiling for chronic wasting disease. And so this was like an experimental infection, and they compared it to naturally collected harvested data, but it seems like this could be something that we could look into in the future. So some of the challenges. There's not a lot of support necessarily for these methods yet. I think a lot of the time we go, well, it might be this and it might be that, and people want a yes or no answer. The sequencing and a lot of these analyses can be very expensive, and our sample collection and preservation is imperative. So it's important that they get into the right buffers, or that they put on dry ice immediately, and sometimes that can be very difficult. But that isn't to say that there are not a lot of opportunities. I am always a fan of bringing citizen science into this, and I feel like if everyone just had a kit on them and found a poop sample, they could just put it in and send it to me, I would be so happy. And that we could get people to collect things on an ad hoc basis and send it in, and if we had some sort of regular testing, kind of like what Crystal was talking about, how they've got regular testing for different things, we could look at this potentially for fecal samples. And just to say that there are a number of people out there, there's hunters, wildlife managers, biologists, veterinarians, wildlife researchers, and the general public who are out in the wild all the time, and we have these people out there that could be collecting data for us. And so in light of this, I put this one in thinking about how could we add wildlife, Anne, back into this participatory platforms, and I was thinking, well, there's iNaturalist, where people are forever going, what snake is this, and taking a photograph, and there's eBird and Merlin for bird data. We could bring these people in and say, why don't we have a thing with like, this is a weird looking thing on the snake? And with that, I will say thank you.